This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only. Check out the new YouTube original channel UCTV Prime at youtube.com/UCTV Prime. Subscribe today to get new programs every week.
Good evening and welcome to the Birch Aquarium. My name is Nigella Hilgarth and I'm the Executive Director here at the Birch Aquarium at Scripps Institution of Oceanography. And it gives me great pleasure to welcome you to the latest Jeffrey B. Graham Perspectives on Ocean Science speaker series. And it's my great pleasure to introduce our speaker for tonight, Dr. Heidi Duer. And I want to start off by saying that this is a very poignant and special evening because I think, as many of you know, Dr. Jeffrey Graham passed away at the end of last year, and Heidi was one of his graduate students here at Scripps, so it's very fitting and exciting to have her carry on his work by talking to us this evening. Heidi Duer is currently a fisheries research biologist here at NOAA Southwest Fisheries, which is literally next door to us here at Scripps. And she's also a leader of a shark working group that is tagging, well, to summarize, it's tagging fish in the Pacific. And she's worked with so many species of fish. I'm not going to list them all, I'm sure she'll talk about them, but particularly tuna, swordfish, and even whale sharks that I'm very jealous of. I'd love to see a whale shark. And she says she's been enchanted by the ocean as long as she can remember. And after she graduated from her undergraduate degree at Oregon University, Oregon State in 1987, she came right here to Scripps Institution of Oceanography to work with Dr. Jeffrey Graham, and she graduated in 1993. And her thesis was focused on the energetics, swimming mechanics, and thermoregulatory capabilities of tropical tuna fish. And then she did a postdoc at the Monterey Bay Aquarium Research Institute, which is in Monterey, California, and she developed tools to address much more complex physiological issues about fish in their natural environment. And then you're moving up the coast. She moved up to Stanford University Hopkins Marine Station, and she worked with Dr. Barbara Block at the Tuna Research and Conservation Center. And she did a lot of tagging of bluefin tuna to look at their movements and behavior, and that provided very critical information for the conservation of that species. And it was actually while she was there that she started tagging mola mola right back here in San Diego. And then she spent 3 years really focusing on applied fisheries work, working on billfish and manta rays. And then she came back to NOAA. And of course, being a good Brit, I loved, when I was a graduate student, eating fish and chips. And in those days, you know, it was card wrapped in newspaper with homemade French fries. And I don't think that's what she's going to talk about tonight. In fact, I know she's not. She's going to talk about fish and chips using high-tech tools to learn a lot more about fish. So please, without further ado, help me welcome Heidi Duer tonight.
Thank you for that lovely introduction. I'd forgotten about half that stuff that I've done over the years. So what I wanted to share with you today is some of the exciting advances in technologies that have allowed us to really take our studies of fish biology to the next level. And I feel incredibly fortunate to be a scientist at this time because I've seen such dramatic advances in the last 20 years. What I thought I would do is tell you a little bit about NOAA, because not everyone knows about NOAA, where I work, the National Oceanographic and Atmospheric Administration. Tell a little bit about the history of fisheries, introduce some of the tools that we use, and then tell you some specific stories about how we've used those tools, and hopefully then use that information to improve efforts for management and conservation. So NOAA is, as I said, National Oceanographic and Atmospheric Administration. We're actually a part of the Department of Commerce. And there are a number of elements of NOAA, but the four main ones that people hear about are, well, first, the main one people hear about is the Weather Service. We actually have our own weather channel, which is very exciting. There's also a lot of satellite data services. You'll see some slides on satellite imagery. So they're looking at everything from chlorophyll or productivity in the oceans, temperatures, how ocean heights, which tells us about currents, all sorts of really exciting things, actually. There's a National Ocean Service, which provides a lot of charts for coastal and oceanographic areas. And then I work for the National Marine Fisheries Service. And as Nigella said, it's right across the street. I work with the Fisheries Resources Division, and within that group, in the highly migratory species group. And highly migratory species are those fish that are capable of traveling great distances. And from a management perspective, that makes them a bit challenging, because it usually means they're not staying just in US waters, but going either into the waters of other countries or international waters. What we try to do is provide the biological data that's necessary for management. And it's actually a different body that actually makes the management decisions. So the species that our group is responsible for, we don't have active research programs on all of these, but we do on quite a few. But the highly migratory species typically include the tunas, billfish, sharks, and then also dolphin fish. And the sharks and white are those that are actually protected. So most of the work that we do centers around fisheries and supporting fisheries, as I mentioned, and also issues associated with fisheries interactions. And in preparing this talk, I did a little research online. It was kind of interesting to learn about the history of fisheries. And people have actually been fishing actively for over 40,000 years. They found bone hooks in Paleolithic middens. They also did looked at the chemical composition of the bones from human remains and found that they were consistent with eating a lot of fish. And there are a lot of examples archaeologically of the history of the importance of fisheries in societies. There you find in Egyptian tombs, there's reference to fisheries. In some of the mosaics from the 1st and 2nd century in the Mediterranean, there's evidence of harpooning for swordfish over the last 2,000 years. And one point that I found really interesting, actually, is that 40,000 years ago in the Paleolithic era, as most people were hunting and gathering and wandering far and wide, a lot of the more permanent or semi-permanent cultures centered around fisheries. So fishing may have been a really important part in a lot of parts of the world for the evolution of societies and developing more complex cultures. And so I kind of look at these fishers from 40,000 years ago as kind of being the first fisheries biologists. Because to be successful, to put food on the table, they would have to know some basic things about the fish that they're eating in order to have a reliable source of protein. So for me, the important questions for those first fishers were likely who they are, who are these fish? And this becomes important because some fish are actually toxic. Like in certain tropical areas, you get the accumulation of toxins in flesh. And so you certainly wouldn't want to bring those home to your family. You would also want to know where you can catch them and if they leave, when they're coming back. So you can make some plans about how you're going to provide resources for your community. And the last two I sort of couch as the understanding the abundance and distribution and temporal and spatial patterns in those. And that's how a geeky scientist like myself would say it today. Well, 40,000 years ago, and through the last 38 or so thousand years, the main focus with fisheries was to put food on the table. In the last, well, it's probably been more like the last 60 or 70 years, we certainly have a lot more people on the planet, and there's a lot more pressure placed on the ocean. So our concerns have gone much far beyond just putting food on the table and have gotten more complex. Things that we now think about in relation to fisheries are certainly fisheries bycatch, because when you're targeting, like we're targeting, let's say, swordfish in a longline fishery, there's an incidence of catch of certain turtles, like loggerhead turtles in Hawaiian fisheries. In purse seine fisheries in tropical waters, There's bycatch of a lot of sharks and manta rays and even juvenile tunas or smaller tunas that you would want to grow up and be larger. Other concerns are overfishing of your target species, and there are some pretty dire examples of situations where populations have gone, are pretty much in the toilet, I'd have to say. Like the Atlantic bluefin tuna, I think, is an example of that, and the southern bluefin tuna is another example of that. So these are the concerns that we have today. And as these concerns sort of evolve, our questions have gotten a bit more complex. So in addition to, those other questions I talked about, who are our fish? That falls under species identification, abundance and distribution, understanding temporal and spatial patterns. We need to know more about stock structure when we're talking about a group of fish, who we're talking about, how far their distribution goes, do they overlap with other groups. We need to look at gear vulnerability. What we do now to try to understand how large a population is we use a lot of fisheries data. And so we're comparing that through time and also through space. So if you have a situation where you're fishing at different depths or your fish are at different depths, for example, let's say in Hawaii you're catching 50% of the available swordfish. Off California you're catching 80% of the available swordfish. Those are apples and oranges. You couldn't compare those numbers directly. So you really need to know how vulnerable your species are to the gear. This also goes to bycatch. You want to know, if you don't want to catch something, you want to know how vulnerable those species are to your gear and try to change that, the catch rates anyway. We need to know about essential habitat, where fish are going and why they're there, because you may manage an area differently if a fish is going there to reproduce or to forage, for example, or if it's a migratory corridor. And then finally, climate change. One of our goals is really to try to be able to differentiate between the impacts of fishing activities, let's say, and environmental change. And we're expecting some environmental changes, and we want to be able to predict how that's going to happen and what's going to happen to our fish. Well, a lot of these questions are not unlike questions that like a terrestrial biologist would ask that's trying to study lions and tigers and bears. But our challenge is that this is the environment that we're working in. You can't see the watering holes. You don't see the babies roaming around on the savannah. You don't see vast areas of forage. So it makes it much more challenging to try to ask those basic questions in this environment. And also, it's not always a very nice place to work. I was actually on this boat, and I'm not the one hanging over the rail, though I've hung over the rail many times in the past, usually for like the first 36 hours. But it makes it very challenging to go out there and do work with these animals, and you don't want to just work with one fish. You want to understand what the whole population's doing. This was actually a trip where we were tracking striped marlin off of Hawaii, and it doesn't look like Hawaii, but it is. So we had a vessel that cost thousands of dollars a day to follow around one fish. And this was state-of-the-art, cutting-edge technology at the time and really important information. The fellow there in the yellow is actually whom I would consider the father of electronic tagging. His name is Frank Carey, and he was a brilliant and amazing scientist. And he actually built the first electronic tags. So this is a picture of Frank inserting a temperature tag into the stomach of a giant bluefin tuna. And that tag is about the size of a Coke can. And this is seriously one of the first electronic tags that was ever built. It was very large. You can see the batteries in there. And in the 60s, scientists, a lot of fish biologists, they would get a tuna and they'd get it on the deck of the boat and they'd put a thermistor in it. It's not a live fish because you can't really do that very easily with a live fish. And they'd measure these high temperatures. It's like, wow, these tunas seem like they're hot. But you can't really say much about what's going on with a tuna that's on the deck of a boat that you stuck a thermistor into. So Frank's goal was to try to take that question and ask it about the fish while it's swimming around happily in these pens that they had off Nova Scotia for bluefin tuna. So he built this acoustic tag, and acoustic means basically a sound tag that would transmit a ping based on temperature. So at low temperatures, it would go ping, 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 and as the temperature got higher, it would go ping, 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 ping. So based on the ping rate, he could tell what the temperature was inside the stomach of the bluefin tuna. And I'm sure this was not easy to do. To me, it seems like it must have been trying to control a child's inflatable pool toy that was attached to an outboard motor, because these are seriously some of the most powerful animals in the ocean, certainly the most powerful fish. So these were some of Frank's first results from that test. And you can see in the light blue, that's the water temperature. And those lines are actually the stomach temperature. And these fish were in pens off Nova Scotia, and they would feed them occasionally. Those feeding events are the little arrows on top. So what you can see is when the fish fed, the temperature dropped, and then after a while, it went back up. And you can kind of think of it as if you swallowed a temperature pill and then ate a bunch of ice cream, the temperature in your stomach would go down. And that's not unlike what it is for this bluefin. With those high temperatures, when they eat something that's water temperature, their stomach temperature is going to go down. And this was the first time that someone was actually able to look at these types of questions on happy fish swimming around naturally, rather than poking a thermistor into a dead fish on the deck of a boat. And if you look at these temperatures, that's 26, so this is about 60 degrees, and that's about 86 Fahrenheit. That's a really big temperature change. And there's, in physiology, often what will happen is as you go up 10 degrees Celsius, function doubles. So let's say that's just 10 degrees. Your muscles' ability to contract would double or more. Your ability to digest would double or more. They also have brain and eye heaters, so their ability to process stimuli would double. So being warm is a huge advantage in the ocean. So anyway, this is kind of an exciting story for me because I like tuna and spent a lot of time with them. But you know, that first tag, it was pretty big, it was pretty simple. It was brilliant for the time. But since that time, we've really come an incredibly long way. I mean, just think about the computers that were available then and what's available now. One of the types of tags that we're using are called archival tags. And this is an archival tag that was in the belly of albacore tuna. And these tags, like their name suggests, archive data. They cost about $1,000, so not too bad. So they're like the little personal computer for a fish swimming around it. And usually in its body cavity, sometimes in its muscle. The limitation to these tags is you have to get them back to download the data. So you can only use them for fisheries where there's enough recovery so you expect to get a reasonable number back. Because if you're going to throw $100,000 worth of tags in the water, you want to make sure you get, a few back anyway. And they're using these on just an incredible number of different species. Another type of tag, my kind of favorite types of tags are these satellite tags. And the reason I like these is because you can put these on a fish and you never have to see that fish again, and you can get all sorts of really cool data. The two that we use for the most part, this is called a spot tag, and we would attach this to the dorsal fin. You can see that up here. That's a spot tag on the dorsal fin. And when that fin breaks the surface, that tag talks to the satellites. The satellite then sends me a message that, okay, your shark's over here. And we've had these last for really impressively long periods. The downside to these is you don't really get any environmental or behavioral data. The other type of tag, this PAT tag, it doesn't talk to you while it's on the animal, but it logs data and then pops off, let's say 6 months later, and then downloads a bunch of data to satellites. So from this type of tag, you can get data through the satellites. So it's a really nice combination of technologies. And so from the archival and the PAT tags, typically the type of data that we get is temperature, so these two, temperature, depth, and light. And from the spot tags, you get a really nice, accurate location. If we think back to our questions, we want to know about the habitat and how animals are using it, and temperature and depth are great for that. We can see where the animals are going in the water column because it's a vast three-dimensional space. And we actually use the light data. This is a light stock that would stick through the body wall into the environment to record light. And that from light, we can estimate geolocation. where the animal is, latitude and longitude. And I'm just going to go over that briefly. So this is, let's say, the day in the life of a tuna. This is depth, and this is light. So we have nighttime, sunrise, day, sunset, night again. And so by looking at when local noon is, we can actually estimate the longitude. So if you think about it, from the perspective of a person standing on the Earth, I know that the sun doesn't go around the Earth, we rotate, but the sun goes around the Earth 360 degrees in 24 hours. So let's say, and all the tags have a real clock on them. So let's say we know that the sun is overhead of Greenwich, England at noon, and let's say that the sun is overhead of us at 8 a.m. or let's say 8 p.m. in the morning. So it traveled 8 a.m. in the morning, or let's say 8 p.m. in the evening. There we go. So it traveled for 8 hours. So that's 1/3 of a day. Well, we know that in a whole day it's going 360 degrees, so that we know that in 1/3 of a day it's going to go 120 degrees. So there's our longitude. It's pretty straightforward, actually. It's a little more complicated than that because the orbit's not exactly circular, but that gives you the general idea. So for latitude, we essentially use the day length. If you know what day it is and you know how long the day is, you can kind of figure out how far you are away from the equator, because day length changes as you move from the equator to the poles. Okay. We play other games to try to improve latitude, because those light-based estimates aren't great, which is why these tags are so awesome, is because we get really good locations from these. So another kind of cool technological advance is come about through cell phone technology. Whereas most of this other stuff has sort of directly benefited scientists, now the public can actually kind of get involved in our science and see what's going on pretty regularly. This is an app that the MCSI put out recently, and it's for iPhones. They don't have a Droid one yet, but they have a bunch of white sharks that they've tagged off of Guadalupe Island. So you can get this app and then go to a fin and then click on the fin and see who this shark is, and you know that that's where it is. So it's pretty cool, actually. And it's really exciting to see it kind of go to the next level. And I think bringing the public in is just critically important, because so much of what we do from conservation and management perspective, we need political will for. And so you need people out there who understand and are kind of vested in cool marine animals, and they're all cool. Okay, so on to some of the stories from our research. First, I'm going to talk about mako sharks. And mako sharks are, they're local, and they're important both in local commercial and recreational fisheries. And they, like our bluefin tuna, are also what we call endothermic, or warm. So that makes it interesting to try and see what they're able to do because of that. So the way that we tag, and this is how we tag makos and blue sharks and thresher sharks actually, is we go out on a large vessel. This was the David Star of Jordan that got decommissioned, unfortunately. We put out a long line. We let it soak for a few hours, not too long, because we don't want to stress the animals out too much. Then we bring them up onto this cradle, and that cradle is hoisted up out of the water. Then we get down there on our hands and knees and attach the fin to the dorsal fin, or attach the spot tag to the dorsal fin, put one of these pat tags in the mussel, and you can see that, you know, it's kind of surprising. This is a pretty good-sized shark, and he's pretty much just laying there. And this, most of the time they do this, actually. We've got a hose in his mouth, so fresh water's going over his gills, and we also put a blindfold over their eyes. And I've done actual surgery on bluefin tuna up to 500 pounds. And when fish are out of the water and often even on their backs, they're actually pretty calm. So it's not as hard as you might think it is. Certainly always exciting. So anyway, with the mako sharks, we've put out 76 of these spot tags, my favorite tags, and been able to document their movements through multiple years, actually. We have an average of almost a year-long track, and 27 tracks that are over a year, which is really important for looking at seasonal migrations. And from a graph like this, you can see that they spend a lot of time along the coast of California, But in the winter and early spring, a lot of animals will come down to the subtropical convergence zone before coming back. And from a management and conservation perspective, if we want to manage those local mako sharks, we know that we need to at least talk to Mexico. And we know that if we're concerned about them, that at certain times of year, if there are fisheries out here, that they may overlap with those fisheries. Because we've gotten a number of multi-year tracks, we can also look at the movements at even a higher level of resolution. And this kind of is some of my favorite stuff, because this is, we didn't really expect this, because you see that big, broad spread of spots, and it seems kind of random with some seasonal patterns. But these are consecutive years of the same individual. The red is 1 year and the yellow is another year. And these individuals went back to pretty much the same location in subsequent years. So it's like they have neighborhoods that they like to go to. And this kind of information is important because if you think about managing, you may want to manage for behavioral variability as well. If you were to fish really heavy in the, well, if you're thinking about like the population level as a whole, and you think that you've got all those points going around all over the Pacific and they're randomly distributed, you may think you could fish more than, like in the Sea of Cortez, for example, than you really could, because if you take out all these few behavioral animals that like to go up into the Sea of Cortez, there may not be any more sharks in the Sea of Cortez, even though there are lots of sharks out farther in the Pacific. So that kind of stuff is important to keep in mind. And what's been kind of fun is that the more we look and the more we get these multi-year tracks, the more we see this sort of pattern. Here's another example from this top program, Tagging the Pacific Pelagics. This is the spread of all the salmon shark tracks around the Pacific. This is our 3 consecutive years for one shark, a tag that lasted for 3 years. That shark went pretty much back to the same location. And you see the same thing. Some sharks like the coast, some sharks like it out here. But that's important information for management as well. Also, because we've been doing this for such a long time, we can compare tracks across years. So the red was the summer for tags deployed in the summer of 2003, yellow is 2004, and green is 2005. And notice how much more broadly distributed those 2004 animals were. That was an El Nino year. So, you know, it's important for us to understand if we see changes in distribution, how they're linked to environment, how they're linked to fisheries activities. And this gives us some insight into what these mako sharks are going to do in the face of environmental change over a short term and possibly also over a long term. As a part of this larger project, they also tagged California sea lions. Sea lions did the same thing in this year. Their migrations were much broader. And this has implications for energetics, too. If you have to swim way up to Oregon, Washington to get some food, you might not be able to invest as much in other activities, like for the sea lions anyway, for reproduction. All right. So we also have been tagging thresher sharks and blue sharks. And thresher sharks are actually the largest shark fishery off our west coast. And their populations declined pretty dramatically in the late 70s, early 80s when they were targeted in a fishery. But the data that we have now suggests that populations are actually increasing and they're doing well, which is exciting. It's always nice to see, you know, some good news coming from the ocean. And then, so we're interested in thresher sharks because of the fisheries, which include both commercial and recreational fisheries. Blue sharks, while we don't target them, they are targeted in Mexico. They use them in their fish tacos. And we catch a lot in some of our local fisheries for swordfish, for example. And they're one of the main bycatch species globally of sharks. And there's mixed reports on how their populations are doing. So we want to know more about the blue sharks. So if we look just geographically, these are pop-off locations using this type of tag for thresher sharks. These tags don't work on threshers. We've spent a little money figuring that out. So here are the pop-off locations where the thresher sharks were. California current, California bite, same place as our mako sharks and our blue sharks. They all really like the California current and the California bite. It's a very productive eastern boundary current. Well, remember, our tags also record depth. And the whole story is not just looking at geographical location. We also have to look to see how they're using the water column. So because we've put these tags out on all three of them, we can start to look at vertical movements. So this is, I think, a week in the life of a mako, a thresher, and a blue shark. This is nighttime, and then the white parts are daytime. This is temperature and then depth. You can see the mako shark likes to spend a lot of time at the surface and then makes these sort of occasional dives down with not too much of a pattern day-night. The thresher shark, on the other hand, likes to spend the days at depth and the nights at the surface. Days at depth, nights at the surface. Then our blue shark is doing these pretty impressive dives pretty much all day long down to about 300 meters. And in order to kind of take this to the next level, these tags don't tell us when the animals are feeding. Our lab also does a lot of work on foraging ecology. So by looking at the stomach contents of the three species and then looking at the ecology of the prey, we can get a better sense for how the depth data sort of translates into habitat use and essential habitat. So our thresher sharks like anchovies and sardines. They like small schooling fish. It's thought that the small schooling fish are easier to whack with their tail and to feed on, because that's how they feed, is by whacking fish with their tail. Our mako sharks kind of like more surface stuff, maybe diving down a little bit. And then our blue sharks, they like to go pretty darn deep. They eat a lot of cephalopods or deep squid, and they forage on what we call the deep scattering layer, which is this deep layer of squid and crustaceans and fish that moves up and down every day, being deeper during the day and then coming to the surface at night. And now armed with this kind of information, we can make better predictions about how our animals are going to respond in the face of different changes. So for example, if something happens with the sardine and the anchovies, we would look to the thresher sharks and see if it's impacting the thresher sharks, because you would expect it to impact them more. Now the blue sharks, they're feeding really deep. And so something that might affect their ability to go down in the water column could determine whether a habitat is good foraging habitat for them or not. So for example, off California and along the west coast down into Mexico, there's a layer of water that has pretty low oxygen that's reasonably close to the surface. So if that oxygen layer comes closer to the surface, it may not have enough oxygen in it for the blue sharks to get down to feed on the animals they would normally feed on. So if we see a shoaling of that oxygen, what we call the oxygen minimum zone, that could impact blue shark foraging habitat where you wouldn't expect it to impact mako or thresher sharks as much. So that kind of information is really important, again, for sort of helping us to understand what patterns we see out there. So another project that we have ongoing is with albacore tuna. And for albacore, we use these tags because we can get them back. We don't get as many back from albacore as we do from some other species. Like the bluefin tags, they're getting about 50% of those back when they put tags on bluefin, which honestly is probably not such a great thing for the bluefin. But the reason we're studying albacore is because they're the most important fishery for highly migratory species on the West Coast. And we don't really fully understand their stock structure. There's a fishery off of Oregon and Washington, and then a recreational fishery down here. And there's been some question about whether that's one group of fish or two group of fish. And the answer to that question is going to determine how you would manage them. So they put out hundreds of tags and got back 20 and found some really diverse and pretty impressive movement patterns. Tags were deployed both north and also down in the Southern California Bight. Some fish, well, this one fish went all the way over to Japan before it was captured. And these kind of show you the seasonal cycles, north, central, north. So that's this group went out to started in the north, went to the Central Pacific, and then back. And then there's some that started in the north, went to the Central Pacific, then went south, and then south, central, south, and then some that hung out off of Baja. So there is some movement between those two groups, but there's also a lot of fidelity to these different foraging areas. The most fun thing about these tags when you get them back is you get that temperature, depth, and light data like every minute. So you have this just phenomenal volume of data that allows you to look at habitat use. So here is the habitat use in 4 different areas. This is depth, and then the color shows you temperature. You can see that in these offshore areas, the animals are going really deep in the water column. And in the nearer shore areas, they're much shallower. This is going to impact their gear vulnerability to in different areas, like we talked about before. So we know if there are fisheries in these different areas, whether or not they may, come across them or be at the same depth as the gear. It also tells us a bit about how they're using the habitat, and with other information from those areas can help us to understand maybe what they're foraging on. All right, now I'm going to talk about swordfish, which I've been working on for quite some time now. They're the second most important fishery locally, and they also have an interesting physiology. They have a brain heater. So they have, every time their tail moves, their head moves. It's just physics. And every time their head moves, they want to compensate. They don't want their eyes going back and forth like this, because it's really hard to focus on a prey if that's happening. So every time their tail moves, their muscles contract to make sure that they can focus at a certain distance. One of those muscles that contracts every time the tail moves is modified into a heater organ, so it doesn't make the eye move anymore. There are other muscles that do that, but it actually just generates heat. So they've got this lovely heater behind their eyes, under their brains, keeping them warm so that when they're foraging at depth, on squid or whatever it is, in cold water, they're functioning just like they're at the surface. And their brain and eye temperatures can be at least 10 degrees Celsius, or like 20-ish degrees Fahrenheit above ambient. So I think they're amazing. Another thing with swordfish is that bycatch issues these days are really shaping how we manage them. So we're not managing them because we're concerned that they're over-harvested. We're managing them because we're concerned that the fisheries are taking too many protected species, like turtles, for example. Turtles are a big one. Loggerheads in the Central Pacific, and leatherbacks locally. This is leatherback. They're beautiful. So the work that I've been doing over the last 10 years or so has focused a lot on looking at the habitat use of swordfish so we can figure out how to target swordfish and avoid turtles at the same time. This shows 4 days in the life of swordfish. This is depth again. This is a day. And this is temperature. So just like those thresher sharks we saw, they're going deep during the day and then coming up night, deep during the day. And occasionally they'll come up to bask. They spend about 8% of their time basking. But other than that, they're pretty much down there during the day. Interestingly, these depths, you see these depths are at about 300 meters. This is pretty typical for close to the coast. As you move offshore, data is for this tag here, those depths increase dramatically, and they're going much deeper in the water column. To try to get a sense for how oceanography influenced depths during the day, actually worked with a bunch of other scientists. There are like 30 people on the paper that came out of this, to get data from all sorts of oceanographic areas. So we've got nice tropical areas, we've got our more temperate California, and then super tropical areas off of Florida. And by pulling the data together from all those different really diverse oceanographic regimes, it gives you an opportunity to get a better sense for how physical oceanography is influencing their movements. And the cool thing that came out of this was that this is the daytime depth of our swordfish, and this is a K490, which is kind of a term that tells you how murky the water is. If you have a higher value, the water's murky. If you have a lower value, the water's clear. So what you can see is that in coastal murky waters, swordfish are much shallower than they are in more oligotrophic or tropical waters. And so then we can try to use this to see how we would target swordfish while avoiding leatherback sea turtles. So currently, the way swordfish are harvested, whether it's by longline or by drift gill net, primarily is at night in surface waters. And that's where the turtles are. They're air breathers. They're mostly going to be, especially in coastal waters, going to be close to the surface. So our idea is that maybe we can target them during the day when they're deep and the turtles are up at the surface. So that's kind of the motivation behind collecting all that biological and behavioral data and then linking that to oceanography. And one upside to the fact that they're closer to the surface near the coast is that they're actually within striking distance of a lot of fishing gear, whereas off of, if they're down at 800 meters, it's really hard to fish effectively at 800 meters. So coastal environments may provide a good opportunity to get swordfish while avoiding turtles in the surface waters. Okay, the last little story I'm going to tell you about are basking sharks. And basking sharks are the second largest fish in the ocean. They get to 30-ish, 40 feet long, not quite as long as a school bus, but pretty close. They have gill rakers that look a bit like a comb, very itty-bitty teeth. So if you see one of these in the water, don't worry. I would actually get in with them quietly because they're amazing animals. And like the largest whales and rays, they're filter feeders. And so their food are swarms of copepods that are about the size of a grain of rice. So they swim with their mouths open, filtering out vast volumes of water to get large numbers of these copepods. So it's not one copepod that's food to a basking shark, it's a huge swarm of copepods that's food to a basking shark. Globally, they're distributed in more tropical and temperate waters, although we're finding that they are crossing the equator. And along our coast, they've historically been seen from like Monterey-ish south, though not really off of San Diego. There was one sighting in this historical data off Baja, but nothing for San Diego at all. They like the Santa Barbara Channel. Another area of high abundance were the fjords off of Canada, and they would see hundreds of basking sharks in these fjords. The problem with that is the fjords were also where the salmon were coming back, and the fishermen wanted to catch the salmon. And so the basking sharks would get caught in the fishing nets, and the salmon fishermen didn't like this very much. And so what happened was the Canadian government, and they're not proud of this, we're working very closely with the Canadians now, but they put in place an eradication program. So they put these big blades on the front of their boats, and they ran into the sharks to kill them so they wouldn't get caught in the nets. And they certainly killed over 1,000 animals this way. We had a couple fisheries, like around World War II, there was a shortage of vitamin A, and shark liver is a great source of vitamin A. So there were fisheries going on off our central coast, and they would use a lot of the animal, but they also used the liver for vitamin A. So between this eradication program, our fisheries, and bycatch and other fisheries, populations, best estimates are the populations have really declined. Unfortunately, there hasn't been like a systematic study of population numbers because of kind of the sporadic nature of the fisheries. And this data is seriously anecdotal, so that's the caveat. But they used to see hundreds and thousands off of California and Canada, and now we see a few. So the best guess, and it's not like there's someplace else in the ocean where now they see hundreds of thousands of basking sharks. There isn't. So the best guess is that populations are actually probably pretty low in comparison to historic levels. And fishing hasn't been going on for quite some time, but there doesn't seem to be a large rebound in the population. Also, although recently we've seen more animals. And basking sharks have very low reproductive rates. It takes them very long to reach reproductive maturity. So they're similar to marine mammals, and they can't handle much of a harvest. So that probably plays a role in a lack of a huge rebound in their populations. Also, there's concern about ongoing mortality, whether it's in local fisheries, because they do get caught in nets, they get caught in trap lines. If it's there, they'll get caught in it, it seems. And there's also the Asian shark fin market, and basking shark fin, one fin went for $57,000. So that's a pretty high incentive if someone sees one on the high seas or anywhere to take it and sell the fin. So because of this whole reduction in population and challenge in trying to develop a recovery plan, because we know so little about them, we started a research program a few years back. And one of the big questions is, you saw the band across the Pacific, and you saw those little spots along the West Coast. We really had no idea how those were connected, and whether animals that, occurred off Central California went up to Canada or not, and when they're not here, where are they? So we started using these towed satellite tags, and this tag actually has a little GPS sensor in the top of it, which gives you super accurate data. So far we've tagged 3 sharks. One shark that we tagged off San Diego went up to Morro Bay, and the tag came off early, which was disappointing. But we did get some pretty cool data in the meantime, and can kind of get a sense for how they're using the coastal habitats. The coolest thing is a tag that came off on February 2nd. This is hot off the presses, off of Hawaii. This is an eight months worth of movement. I don't know where it went in between yet, because the tag's actually still transmitting us data. But that shark went from San Diego to the waters near Hawaii. So that tells us that these animals are going to the Central Pacific, and they may be going to Japan. The next tags we deploy we'll try to put out for longer. It's nice to get some good data before you really push it and get really long deployments. This is the depth data that has come in so far. This is the minimum depth in a day and the maximum depth. One thing you'll notice is that as this animal moved offshore, it was going a lot deeper. So similar to our blue shark and our swordfish. But the interesting thing is they're not necessarily coming to the surface every day and spending all of the day at 115 meters or below. The one upside for that is there's a deep set longline fishery for big-eye tuna out in the Central Pacific, but not much going on from a fisheries perspective. So I would think they would be pretty safe at these depths out in the Central Pacific. It's probably, if they are going over to Japan, they'll probably come back up into shallower waters when they get over there. So because they're pretty infrequent, we started an educational program, an outreach program. So if anyone sees a basking shark, you can email the basking shark at basking.shark@noaa.gov. Or you can make a phone call. And we've actually got we're starting to get more reports, which is great, because they're so rare that we can't depend on being out on the water looking for them. We really need the help of all the other people that are out there. The last thing I wanted to mention was a really large tagging program that has added multiple orders of magnitude of information on movements of animals in the Pacific. It's called Tagging in Pacific Pelagics, and it went from 2000 to 2010, and it's evolving in different ways now. But over that 10 years, they tagged 23 different species. And whereas before this project, you might have had a few scattered dots for different species around the Pacific, now the Pacific is pretty much covered. With the tracks from albatross and turtles and whales and shearwaters and tunas and sharks. And it's, from a management perspective and a conservation perspective, this is incredibly powerful, because where we're really going is using more quantitative, complex modeling approaches to try to predict in time and space where you're going to have overlap between species you don't want to catch and species you do want to catch, and trying to minimize that overlap. You also want to maximize your fishing efficiency. So this type of information puts us a huge step forward in those sorts of efforts. And you can see actually a lot of this data if you go to www.top.org. It's still up there, and there are an amazing number of publications and some other things there. And these are my 2 beautiful children who were sad they weren't going to be on TV with me, so now they'll be on TV. So thank you, and I'll take any questions. So the question is, we're collecting temperature, depth, and light data, and what other data is feasible to collect? There actually is a whole explosion of tagging technology, and people are now putting special light filters on the tags so that they can measure chlorophyll, not just at the surface but also vertically, because there's a lot going on below the surface that we can't see with satellites. So that's one example. We at Fisheries were working on an acoustic tag that would not set off a ping so you could follow it, but actually would give you a sense for what prey was around your fish, or your shark, or so it would like a fish finder, basically, a fish-based fish finder. There's a tag like this that's being developed to record the Earth's magnetic field, because we're always trying to improve our location estimates, and that's another way to sort of pinpoint your location, because the magnetic field changes in a known way around the planet. So those are, and there's accelerometers, there's speedometers, there's really, if you can think of it, they're probably putting it on a tag. Inclinometers, all sorts of crazy things. But one of the cool things that a group has been doing, I think they did this work in 2006. They put a stomach pill inside a sea lion, and that pill transmitted to a satellite tag on the back of the animal through the animal. So then they could figure out when the animal is feeding and then look over its range and say, okay, this is important foraging habitat, this is where they're migrating, and this is where they're reproducing, which is just incredibly powerful. How much bias is introduced by where you tag the fish? That's been a challenge, and people are trying different modeling approaches that I don't pretend to understand to deal with it. I mean, the thing is, the point being that if you tag a bunch of fish in the Southern California Bight, how representative is that of the population of fish? And the biggest challenge is, at some level, we can't get around that, because we can't go out to the Central Pacific and try to tag a bunch of those albacore. There just aren't the concentrated fisheries out there, and we don't really have the money or the time to do the perfect random tagging experiment, which we realized would be ideal, but you do the best you can with what you have. So some bias. So the question was, for the tags that we have to retrieve, the archival tags, who they come from and how they know. There are two ways to get tags back. One is you need a really large education and outreach program, and two is you need a large reward. So seriously, there are studies that have shown the higher the reward, the more tags you get back. So there's a project in the Atlantic called TAG on Atlantic bluefin tuna that's run out of Stanford. They offer $1,000. And even with $1,000, it's sometimes hard to get tags back. And part of the problem is that the fishermen feel like if there's too much information, we're going to shut them down. So that's something that you have to combat. Locally, the rewards for these sorts of tags, I think, are about $250 to $500. So a large reward, and then a lot of education and outreach, and focused on commercial fishermen and recreational fishermen. Also, these tags have address information on them and phone numbers, and we have them in, some have them in Japanese and Spanish, and depending on where we're putting the tags out, like this one has it in Japanese. So Sarah was asking about the blue shark's vertical movements, where they would go down deep and then come back up to the surface, and it looked like what you would expect to see from a marine mammal that had to come to the surface to air breathe. Frank Carey, the father of electronic tagging, who you've all met earlier, he did some really nice studies where he looked at the body temperature of the sharks and their vertical movements at the same time. And what he saw is that they would allow their body temperature to fall so low, and then they would come back to the mixed layer to warm back up before they could go back down. Because remember how important temperature is for function. So that's one of those situations where if you're endothermic, like the mako shark, and like our bluefin tuna and our swordfish, you're in an advantage over those fish that aren't, because you're not going to have to do that nearly as often, so you can stay down there and forage.